The session is led by Allison Taylor, a scientist who is doing her part to end CMT for herself and for her family. Allison is a neuroscience graduate student at Johns Hopkins, and she studies axonal CMT using fruit flies, and she is living with CMT1X. She holds a BS in biology from the University of North Texas, and she's conducted research at Oregon Health and Science University. Allison's going to moderate a panel of biotech representatives to discuss their CMT projects, the challenges, the progress, and the patient involvement opportunities, and which really reflects her dedication to both the research and to advocating for people with disabilities. So, Allison?
I'd like to invite my panelists to come up, and while they're getting seated, I will do a little bit of an introduction. First of all, I'd like to thank the CMTRF for inviting me to moderate this panel. It is especially an honor as a patient and a scientist. And my hopes for doing this is to engage the patients and talking with the people that are making the therapies that will eventually make it to the clinic. So our panelists today provide insights on cutting-edge projects and the challenges that they face, the progress that they have achieved, and opportunities that may still lie ahead to combat CMT. With the wealth of experience and diverse perspectives at the table, they will offer insights into research and treatment and emphasize the critical role that us as patients play in advancing these therapies. I will now introduce a few, all of our, not just a few of them, all of them. Okay. Dr. Tim Pizer, if you'll raise your hand, thank you, is a neuropharmacologist with over 25 years of experience in neuroscience and drug discovery. He's held leadership roles in both the public pharmaceutical companies and startup biotechs. His career includes leading drug discovery programs at AstraZeneca, including work on treatment-resistant depression and phase 2 clinical trials. He's also contributed to the development of NMDA receptor modulators for depression and schizophrenia and SK channel modulators for spinocerebellar ataxia during his tenure as CSO at Cadent Therapeutics and as EIR at Atlas Venture, which ultimately led to the Cadence acquisition by Novartis. Dr. Pizer earned a PhD in pharmacology from the University of Minnesota and currently serves as CSO for Samsara Therapeutics. Next, we have Dr. Rachel Salzman, who we met a little bit earlier today. Dr. Salzman brings an extensive experience in the biotech sector, having worked at Allison Therapeutics and co-founded Swan Biotherapeutics. Her dedication extends to addressing ultra-rare diseases through her nonprofit organization, Ultra Squared Bio. Dr. Salzman's background in veterinary medicine and her leadership roles in various biotech ventures make her a valuable contributor to the field of gene therapies and rare disease treatments. Dr. Salzman serves as the global head of corporate strategy at Armatus Bio. Next, we have Dr. Natalia Sacilotto. Dr. Natalia Sacilotto is a molecular biologist specializing in epigenetics and its role in human disease. Her work at Oryzon Genomics, particularly in the developing precision medicine approaches to conditions like CMT1A, demonstrates her commitment to innovative therapeutic treatments. With a strong academic background and translational research experience, she plays a key role in bridging laboratory findings with clinical applications. Dr. Sylvain Celanire, perfect, with over 2 decades of experience, well, it's a little more than 2 years, 2 decades of experience in the biotech and pharma sector. Dr. Celanire is a seasoned executive director, entrepreneur, and advisor. He has played pivotal roles in industry academic partnerships, drug development, and clinical trials. His extensive patent portfolio and research contributions underscore his expertise in advancing drug discovery programs. Currently, Dr. Celanire is the Chief Executive Officer of Augustine Therapeutics. Dr. Christian Lorson, Dr. Lorson is a distinguished scientist and academic leader with a focus on neurodegenerative disease. His career, spanning both academia and industry, has led to significant advances in our understanding and treatment of conditions like spinal muscular atrophy, CMT, and ALS. His multifaceted role as professor, researcher, and company co-founder highlights his dedication to improving the lives of those affected by rare diseases. He serves as the Chief Scientific Officer of Schiff Pharmaceuticals. Did we get everybody? Thumbs up, awesome, okay. All right, so you can tell by the introductions that we have an extremely qualified panel here. And I'd like to try and get us back on track on the schedule. So if we don't answer all of the questions, then that's okay. And I'd also like to open up the opportunity for more questions from the audience. So my goal is to end right at 2:45. Is that, okay, great. All right, so first question, and this will go to all panelists, please. Will you please give a brief 1 or 2 sentence explanation as to what you're working on and why you are working on CMT? Maybe we'll start with Rachel. Yeah. You have to press. Oh, yeah, take it.
So at Armatus Bio, we're working on vectorized RNAi. In other words, we're targeting autosomal dominant diseases. So that means the pathology is a result of too much of a particular gene getting expressed, and we're delivering microRNAs that are going to silence that extra protein so that it doesn't get expressed. The reason that we're in CMT1A is because it's probably the quintessential example of a rare disease where this type of technology should be effective. And already we've seen, as I alluded to earlier this morning, that at least based on our mouse experiments, so when I are alluding to our scientific partners that Edred's called out, Dr. Cleopa and Dr. Harper, that in the mouse models, this is showing to be the case, that we're able to reduce the levels of PNP22 to therapeutic levels. And the other part that's appealing about CMT1A, besides the fact that I personally have had my eye on it for about 4 years, is that it's a very common rare disease. And there's a tremendous amount of natural history available that's been conducted for years through the Inherited Neuropathy Consortium. And I think in terms of rare diseases, the resources in terms of clinical trial readiness that the INC is able to provide through probably many people in this room's cooperation and participation really does differentiate not just CMT1A, but all CMTs and other inherited neuropathies. So we see this as a really, really compelling opportunity.
Hi, so Augustine Therapeutics is a small biotech company created 3 years ago. We are developing new small molecule drugs that target HDAC6 inhibition as a mode of action to restore axonal damage in many subtypes of CMT. The reason that we are there is that, in fact, Professor Ludo van den Bos, the scientific founder of Augustine, has been the first to demonstrate 10 years ago the role of HDAC6 in CMT2A, CMT2F, and CMT2D. And we extend with Augustine into CMT1A. And so this is where we are now. We are, we say, CMT agnostic subtype. We are trying to solve all of them. And we start with CMT1A at first.
Samsara Therapeutics, led by didn't have that on. Indeed, Samsara Therapeutics, which is led by the CSO, Peter Hanley, head of biology, John Blackwood, Warren Galloway, head of chemistry. That team is very familiar to the CMT-RF. And they have developed a method to identify new small molecules that promote a fundamental cell process called autophagy. Autophagy was the subject of the 2016 Nobel Prize in Medicine. And it's the way that cells engulf and discard damaged and excess proteins. It's been linked by scientists to 18 of the genes that are causal in CMT. Our work so far has been in CMT1A, some of the models that Rachel described. And so far, the team has shown that, that these compounds delivered at different doses and over a month of timer showed to these mice actually improve their function. These mice develop, as you know, uncoordinated movements, deficits in nerve function, changes in the nerve structure. And we've seen, as Rachel pointed out, for compounds, the target here is to reduce PMP22. This autophagy process appears to do that. It's induced in the Schwann cells, and the mice show improved function, improved nerve conduction, and we're developing these compounds as treatments for CMT. Thank you.
Good afternoon. Chris Lorson, and Steve O'Connor and I co-founded Shift in 2017, and Shift Pharmaceuticals focuses on antisense oligonucleotides, or ASOs. These are short synthetic stretches of nucleic acid, and it's essentially a gene therapy, but not within a vector. So these ASOs can do a number of different functions, but the particular one that we're interested in is modulating pre-mRNA. So it's the first step after goes DNA to RNA, and that first RNA is called pre-mRNA. It's basically a copy of the DNA, but in the RNA molecule. And we are changing the way the chapters are essentially read. So we induce a skip. Instead of targeting the RNA for degradation, we let the cell take care of that by kind of, screwing it up, basically. That's kind of the clever way to say it. And you see degradation of the RNA. So we're not removing completely PMP22. We're in the CMT1A space. And what we're trying to do is just reduce in a titratable manner using antisense oligonucleotides to reduce PMP22. So, and the way that we got into this was actually Charlotte Sumner, who's been a friend in the SMA space for about 15 years, gave us a call. She said, hey, I work with CMTRF. I'd like you to think about, applying for a grant. And that was the first step. So kind of an aggressive group, and it's great to see so many people here, and we appreciate being targeted.
Hello, everyone. So I'm Natalia from Oryzon Genomics. Just to give you a quick idea of who we are and why we came across CMT. We're a small but mature clinical stage biotech based in Barcelona. And we're fairly new in the CMT field, but we're certainly not new in the rare disease space. We've been developing epigenetic drugs for many years now for diseases with high unmet medical needs. We have 2 clinical candidates in phase 2 in different fields, in oncology, for which we have been granted orphan drug designation for AML, small cell lung cancer. And we have another clinical asset in neuropsychiatric disorders, and we are also focusing on the ultra-rare disease, Kabuki syndrome. And in our pipeline, we also have a really selective and potent and cool HDAC6 inhibitor. And in 2020, we approached the CMTRF because given the well-established role, HDAC6 has, and it has been very well described in mechanisms that are impaired in CMT and other pathologies like ALS and other pathologies. So for us, and I said this to Clary and to Susan and Edrit, to us felt like a moral obligation to test these molecules in models of CMT. And this is why we approached the CMTRF. And thanks to them, we obtained really great data on CMT1A showing efficacy. And, we're really happy for that. And so that's why we're focusing on CMT, thanks to the CMTRF. And we are also interested in other rare diseases. So thank you.
Yeah. On behalf of all of the patients, we are so happy that you are working on CMT. Thank you. And I've noticed a bit of a theme starting in the first session is we're talking a little bit about hope. It seems like it's maybe it's always darkest before the light comes kind of moment right now. Everybody's working towards the same goal, and we're not there yet, but we're feels very close. So I will direct this question to Chris and then Rachel, if you could give us some more hope. What are you excited about in the field right now? And should we as patients share that excitement?
Yes.
Thank you.
So, I mean, I think you've heard SMA mentioned about 1,000 times in the last 24 hours or so, but I think that as a rare disease, you kind of use successes to use those as stepping stones. And I think that SMA was a great test case for rare diseases. And it feels so much like SMA from 10 years ago. Things hadn't really started to hit. There was beautiful preclinical work. I see beautiful preclinical work here. The models seem to be very, very strong for many, and not all, but many, especially 1A, that this is a great setup for translating to clinical success. I think it's important, though, to realize that I think someone said it yesterday, that the clinical trials are not going to just be confirming our preclinical work. These are actual experiments. So we really have to be thoughtful about them. And I think that there's an enormous opportunity here. And to have multiple shots on goal, which Pat said, which is, I think is the perfect way to phrase it, is that there are multiple shots on goal coming from different mechanisms. So if you can address a problem with small molecules, ASOs, gene therapy, and then things that are kind of genetic lesion agnostic, things that help the neuron, help the muscle, et cetera. I think those are platforms that are really powerful. And to see everything moving forward simultaneously, I think is really exciting.
Thanks. Picking up on Chris's last point, it even struck me as I was listening to my co-panelists here, you don't have to pick one of us. You probably can pick all of us. The therapeutics that are already making a lot of progress, even from the amount of data that I'm learning about over the last few days, they're not mutually exclusive. And we're already seeing examples in other rare diseases like SMA, where there are multiple precision medicines, where, as Charlotte alluded to during the keynote, it's not that one usurps the other. It's all about giving opportunities for patients to reach maximum clinical effectiveness. So I really foresee all of these moving parts that are not just multiple shots on goal, but we can actually make our goal very high. The goal is as much alleviation or reversal or improvement of the given condition. So that's first of all. It's always good to have a lot of options, and I do believe they can be complementary, and we're already seeing the examples I'm talking about in SMA are very recent, over the last year or two, and not necessarily what we were expecting. The other reason what I'm excited about, which not to continue to repeat the DTX deal, but it really is important, not just for the science side, but also for the business side and for the patient side. And all of those triangulations are actually very relevant when it comes to contributing to the probability of success of a program, a company, an asset moving forward. So to be able to just give that the shout out that you need in the middle of a conversation, it might be to your investors, it might be to your family, it might be to your colleagues. It actually contains a lot of power. And then thirdly, there's been some recent regulatory approvals that have really very much changed the way I'm thinking about how FDA is literally doing things, not only talking about things. Two recent kind of classic gene therapies, the one in Duchenne's muscular dystrophy and the one in the disease that's in my family, Skysona, those were approved by CBER based on accelerated approval. And from the word, without having to go into a whole regulatory lecture here, obviously it's an accelerated approval, which means patients are able to get access to it sooner than a traditional approval. And FDA kept telling us, we can do this, and they're finally starting to do it. So I also see, I'm excited about not just getting the drugs into the experimental setting like a clinical trial, but I'm actually seeing an opportunity getting these approvals happening sooner than I might have thought of even maybe 18 months ago. So that sort of sums it up.
Yeah, that's very exciting. Thank you. I think another big question on a lot of our minds, and it was alluded to earlier, is most of the companies that have been mentioned today have been mostly working on CMT1A. And myself and my family have CMT1X. And so I'd like to direct my question to Natalia and then Sylvain. And that question is does your therapeutic approach focus on only one type of CMT? And is there applicability to other types?
So far we have great preclinical data on CMT1A, but the mechanism of action of HDAC6 inhibitors obviously does not preclude it from being efficacious in other CMT subtypes. And indeed, HDAC6 inhibition has been shown to improve myelination, to improve axonal transport of mitochondria and other important cargoes, to improve mitochondrial health, to reduce inflammation. And these are all things that most of them are like in pair across CMT subtypes. So we are very confident that with this strategy of improving the overall health of the neuron feeling, healthier and happier with better bioenergetics, mitochondria are the ones that make up energy, right? So if mitochondria are healthy, so the neuron is healthy and happy. So as I said, we have great preclinical data on CMT1A, but we definitely are looking to, focusing on other CMT subtypes, definitely.
Similar, joining what Natalia just said, definitely the origins of HDAC6 for the axonal form of CMT2. Beyond CMT1A, we have now data on CMT2A from patients. So we have been working on iPSC cells that I would say we have a clinical center in Leuven with a lot of CMT patients, and we have access to a patient line. And so we have been demonstrating now that we can really reverse the kind of target engagement tubulin, which is one of the key factors for axonal transport defect in CMT2. And we have now demonstrated also the data on CMT2A. So the goal is also not to stop at CMT1A. We have CMT2A data as well. And with all these patients pool in Leuven, the goal is also to look forward, bringing the solution for this form.
Yeah, that's awesome. Thank you. And I'd like to address the next questions to Tim and then Natalia again. And that is, how has the CMTRF helped you get your project moving forward?
Well, like many of the companies at the table here, and you saw Edritz's presentation about the portfolio, we have a grant from CMTRF, and that helps us to purchase the supplies and reagents we need to conduct the studies. We talked about these mouse models. They're bred by companies that specialize in the breeding, and to access those mice is expensive, and it's a long-term process to test in them. So that's been a key element. I think the other thing I really commend the CMTRF on is the scientific advisory board. So when you put your grant in, you don't just get money back. You get, first of all, a filter. So kudos to the team for picking the best. It will help the field going forward if you apply that winnowing fan. But then you get advice on how to conduct your studies, how to get them optimally tuned up to support the field going forward. That's not just advice for you personally as a company, it's advice for the field. How does this compare to another therapeutic, another modality? Can these be combined together to treat more effectively? This is the kind of feedback that we get as we launch on our efforts. So that's, I think, really helpful.
Awesome.
Yeah, I have very little to add to that because we feel the same way. So we obtained a grant from the CMTRF that basically allowed us to get all this very, important preclinical data, in vivo preclinical data. But it's not just about the funds and the money. We are, receiving invaluable advice. As I said, we are new in the CMT field. We have a lot of experience in the CNS, but we are learning a lot about the PNS. And when our grant has been reviewed, we received very valuable feedback, and we are learning so much along the way. And this is all thanks to the CMTRF, and we're incredibly grateful. And we certainly hope to continue our journey together. And so it's help and advice in every way. So thanks a lot.
Yeah, thank you. And when we're thinking about delivery of therapeutic approaches, we know that certain tactics may be more like hazardous than others, and I know we're taking a lot of safety precautions here. So to address a prominent patient question, I'd like to ask Chris and then Rachel, what considerations are you making in the development of your therapy towards safety whenever it finally makes it to the clinic?
So the particular compound that we're working with, the synthetic nucleic acid, it has a backbone, and the backbone is referred to as morpholino. And this is something that Sarepta has been injecting for a decade and a half. You can inject an enormous amount of these compounds, and the backbone intrinsically has almost no activity. So it's very, very inert. So from that side, the simple structure of the molecule has very little toxic or side effects. It will allow us, I believe, to really ramp up the amount that we can inject. And so I think that being able to dose high to kind of drive entry into particular cell types, Schwann cells, will be really important. And so what I also find appealing about our particular approach is that it's titratable. So you don't want too much and you don't want too little, and being able to have something that is essentially manageable and titratable with this ASO approach, I think is really important as well.
Can you explain that a little bit more, titration in that regard?
Well, I think that finding the appropriate dose allows us to, and that's part of the safety studies that we'll perform, is to find the appropriate dose that lowers the PMP22 to essentially a Goldilocks level, right? You don't want it too high. You don't want HNPP. So I think being able to adjust the dose is going to be critical. And I think that's something for long-term dosing as well. Just being able to manage and view how the drug is acting over time will be really important. And so being able to adjust the dose up or down is critical.
Thank you. Rachel?
I really appreciate getting this question. So when I mentioned that we have a platform where we vectorize RNAi, the vector that we're using is AAV9. So first of all, we're doing extensive preclinical work. So that's in small animals and in large animals. And that, of course, is mandatory and a rational way to do your first steps in terms of evaluating safety. And of course, so far, everything, I shouldn't say of course, but so far, everything looks good. That being said, what gives us added comfort as we move forward with this strategy is the one approved AAV product for a neurological disease, Zolgensma, which is for SMA. They are using AAV9. Therefore, I believe at this point, at least over 1,000 patients have been dosed with this. They're being dosed with much higher doses, and they're being administered it intravenously, and we would be delivering ours more like Spinraza intrathecally to your cerebrospinal fluid. In other words, our total dose is lower than the AAV9 dosing paradigm for SMA. But the point that I want to make is the vector that we're using has already been used in the clinic as part of an approved product. And I think that the value of getting real world data, even though it's a little bit adjacent, but it's still highly relevant, is something that we monitor and keep track of very carefully. Furthermore, in terms of thinking about immune responses that have been reported in some other gene therapy studies that you might have read about, our payload, our cargo, there's all kinds of words for it. It's not it doesn't code for a protein. It's just for a microRNA. So it's not expressing a protein. So it's not going to be immunogenic. And our animal data so far backs that up. So these are sort of different points along the road of making you have confidence in your safety. We're also checking it at different doses because, of course, that's always important. And you want to find that sweet spot dose where you get both maximum safety and, of course, maximum efficacy. And then the other part in terms of taking it forward to the clinic, and that's why yesterday was so helpful understanding the patient perspective. We're going to be sure that, especially when we test this initially in a relatively small group of patients, we think very carefully about who those patients are, where they are in their disease journey, what kind of risk benefit they are comfortable with. We also have to be mindful of what the agency is comfortable with and what makes sense to our own clinical development team, but that must absolutely be anchored to what the patient community is comfortable with. And therefore, by being able to pick the suitable population with all of this package or totality of data alluding to safety both in animals and adjacent in humans, those are the steps that we're taking. And it's an evolving process. We get more data, and that helps us continue to make sure that we're always going towards that sweet spot of ideal efficacy, obviously with safety. I hope that answers it.
Yeah, thank you. And we actually do have the time to add another person to maybe answer this question. Sylvain, what considerations are you making for safety?
So in the case of HDAC6 inhibitors that have been a concern for safety for the first generation of compounds that have been developed so far for oncology purposes, there was some genotoxicity for the third generation class of compound 20 years ago. There was cardiotoxicity that had been also seen, and hematotoxicity in patients. And most of the biotech company and big pharma companies have been moving away from the generation of compounds for, I would say, better drugs, still targeting selectively HDAC6, and Oryzon Genomics and others have been doing that for the past years now. We have been really looking at the safety from the preclinical stage and even from in vitro stage, rather than going to a new class of compound without looking at the safety too late. And so what's in for the pharma industry and what the investors looking at the, I would say, at the project, they ask, are you different? How you differentiate yourself compared to the third generation, the second generation? Are you better on many cases? And so the goal is to screen early the safety in vitro key tests that are, let's say, relevant and also required by the agency. So we did that for our clinical candidates upfront. Very early on, we look at in vitro, that was clean. We moved to in vivo. The non-GLP tox data are very clean now. So now we can move to regulatory GLP tox studies to be sure that we can advance this compound to adults, but also for pediatric use. It'd be important for children to have such drugs. So we have been very careful for this particular generation of compound that have been facing issues in the past.
Yeah, awesome. Thank you. Next to Tim and then Natalia, if you could explain to us what stage are you at in your research, and how long do you think it will take to get to a therapy in the clinic where you're at currently?
Measured on the scale of a human lifetime, drug development is slow, especially if you're living with one of these conditions. So I understand that. And the kinds of safety testing that Sylvain just described seems heavy load until it breaks down, and then it's absolutely necessary. So right now, Samsara has molecules that are poised to go into the clinic and other indications, but our efforts in CMT are a little bit early stage. We're doing optimization to find the best molecule to take forward. And then, of course, we'll do all of that safety testing we just described before we take it into the clinic. So it'll be still a little while longer.
Yep. So we are at the stage of, so we have nominated a candidate and we are now in the phase of IND enabling studies. So it explained before what it means. Those are all the studies aimed at making sure that your drug is safe, it's non-toxic, and it's efficacious. But if it's efficacious and it's not safe, you know, it's worthless. So these studies are basically aimed at understanding whether your molecule is safe. And we are now performing those studies, and we estimate, fingers crossed, that we could be in first-in-man trials in around 18 months. That would be in healthy volunteers. And we estimate that we could be in CMT patients in 2026, by 2026. But drug development is slow. It takes time. It's a long run. And so from here to commercialization, we can expect something like maybe 10 years. Okay, so this is something for patients to consider. So you have to be happy that this is, a really great progress that we're making, with the awareness that this is a long run, but there will be winners. I'm really sure of that. So yes, we really hope to arrive really soon.
Yeah, that's awesome. Thank you. And amazingly, we are good to go on the last question. We're actually going to end on time. I'm so proud. And so this will be addressed to all the panelists. We'll just go down the line. Currently, there's a room full of patients here in person, but also online virtually. What's up, Rob? And we want to ask you, what does it mean to you and your company to have the patients be involved in the process, to be here to talk with you? And then whenever y'all eventually say, oh, it's awesome, like a bunch of times, then you can maybe answer a third question, which would be, do you have a message to give directly to the patients? Thank you. Yeah.
I think the message to the patients is we need to continue this form of partnership and communication. It's very important, and this is a personal thing. I'm not speaking on behalf of my company now. I don't want you to ever think that or feel that you're being patronized. Are not fully empowered as partners in this. That is what this is all about. And I look forward to learning more and then being able to, I don't think the right word is weaponize, but maybe it's activate all of this information that we're getting to make sure that our team, first of all, moves forward quickly and appropriately, and that we can also use that progress to share information with the regulatory agencies to educate them in advance so that they know all of these CMT1A therapeutics are on a train coming their way, and they should be fully prepared to have all the information they need in order to, if appropriate, approve those drugs. And this is where the patient participation and partnership is going to be critical. I've been to many FDA interactions, and like most meetings, there's always people whispering in the background or doing whatever they do. Every time a patient is speaking, you can hear a pin drop. So I think that that's something that my message to the patients is I want to be able to activate / weaponize it, and we're in this together.
So my message to the patient is very clear. We have a lot of, I would say, as seen by the pipeline, a lot of companies, a lot of breakthrough that have been in science, in development. We have now one big pharma taking over one project that has been funded by the CMT Research Foundation that gives hope that all the other big pharma will take this train towards approval and clinical development, where we have, I say, challenges. We have at Augustine the chance to have a scientist and a patient, which is Rob Pryor, which is down there. He has been at the inception of the company with Ludo, our scientific founder, and by having a patient next door in the lab, knowing how he can be courageous to work in his own disease, but also bringing his voice to what we need to do. In vitro testing, in vivo pharmacological model, the relevance of the outcome measures, the biomarker, that has been instrumental to the success where we are now after 3 years with a compound entering clinical trial next year. So by fostering this collaboration with CMT Research Foundation, we have also the chance to have in Europe the European CMT Federation that are also fostering all the national agencies and also EMA, FDA collaboration. The goal is really to foster that, to strengthen that links, to accelerate the approval of new drugs. So we are not yet there, but we are on the right track.
You know, the clinical indication, the clinical syndrome of CMT has been known for more than a century. And recent genetic evidence has identified more than 100 genes. Ask yourself, what does that mean that more than 100 different genetic mutations can cause the same clinical syndrome? I think one of the things I'm excited about is the possibility that there is an underlying common pathogenic mechanism. The same cells are dysfunctioning, producing the same clinical symptoms. Is there something we can target? Maybe we don't have to go one gene at a time. We can target an underlying mechanism. Obviously, at Samsara, we're hopeful that that's autophagy, but if it's not, it might be something else that could be targeted by another medicine and bring relief more broadly to these folks. Obviously, it's very exciting to see you gathered here today. When you talk about your condition, it sounds like what the FDA needs to hear to make sure that when they regulate the development of these drugs, you get what you want out of the other end. So advocating to them directly is your way to make sure that that happens. Thank you.
It does look like we're on time, so I'm going to tell a story and screw stuff up.
Yeah, go for it.
But what I wanted to say is that being relatively new to CMT, I've been in SMA since 1997, and I wrote an essay for a family. It was for a Shire Brave Award, and I'll summarize it because it was about 3 pages. But the bottom line was that there's a mother. She has 2 daughters. They both have SMA. One has type 2, which is very severe. The other's type 3. And she's doing bake sales. She's doing car washes, fun runs, stuff like that to raise, relatively small amounts of money that go towards developing therapeutics. And one of the lines in the essay was that, she sits there, she goes to bed crying every night, and she knows that she's developing these therapeutics for not her kids, but for other children that don't know that they'll get SMA. And I have to admit, I was 100% wrong. Those therapeutics are in both of her girls, and they're both still alive. And the fact that it went from 0 hope to, holy cow, 3 approved drugs in this incredibly small amount of time, I think is pretty dramatic. So it doesn't happen without kind of a team. And I think that functioning as a team is really important. So I see that in the CMT community, and I think that everybody should really be applauded for that. So it's great to be part of this group as well.
Yeah.
Very little to add. So what I would say is, we wouldn't go anywhere without you. So we need you. We need you to understand what you need. We need you to participate in clinical trials. We need you in every step of the way. And we need you to advocate for yourself and make sure that, you make as much noise as you can, and participate in events like this and do everything you can. And, we need you. So we're super happy and super grateful, that we had the chance to listen to you today and yesterday. And for me, it was amazing. So yes, we're really happy, and I hope that, you keep this energy and this hope forever. I mean, we will get there, I'm pretty sure.
Yeah. Awesome. Thank you so much. And what a great way to end the questioning for the panel is to say, like, it's amazing to have the patients here and to have that interaction. And I'm also so glad that we were able to get through some of the pre-written questions so that we can have more interaction with the people in the audience, both in person and virtually. And these are the people that are making the therapies for us. So if you have any questions, now would be a great time. We have 15 minutes. Yeah. Well, they're bringing you a microphone. Yeah.
I guess my question is, what engagement have you individually or as a group had with FDA engaging about clinical trial designs, endpoint selections? Have you had a research day with CBER and CDER to teach them about CMT? I'm sure they're not that familiar with it.
Mm-hmm. Right. Has anyone had any direct contact with the FDA yet? Yeah, most of these people are preclinical. Yeah, okay. Thank you, Susan. Yeah.
It's such a great question. Thanks for asking it. That's one of the ways that we're de-risking clinical trials. And we don't have it up there because we didn't put any direct funding to FDA for it, but we definitely put a lot of human capital toward it this spring. And in mid-April, we held a listening session with the FDA. And it was an hour long, it was virtual. And we really focused on CMT1A just because that's where we're so close with DTX. But we wanted to paint the picture that it wasn't, you know, the mild version of CMT1A or the type that people don't really notice, because we know that that's the stigma. And so what we did was we engaged with a few people who had more severe forms of CMT1A, legitimate, there's, you know, nothing wacky going on there, but just telling their stories about the loss, the emotional loss, the physical loss, the financial loss of living with a progressive disease. So that was a great meeting. The best news, we also had David Herman there, who's a clinician from University of Rochester, who's focusing on all that clinical trial outcome measures. And so he talked about the state of the field. So that was really great. And then I guess the best news is we had 18 people from FDA there. And we had people from not only the Center for Biologics, which is all the gene therapies, but we also had the Center for Drug, which is all the small molecules. So they have different divisions. They have neurology and in both of those divisions, and they were all there. When we showed the list of attendees to the folks at DTX, they were just astounded at the attention that we were able to garner at that meeting. So we've started the dialogue with FDA. What we learned yesterday in talking with one of the panelists is that you start the dialogue and then it goes from there, that this is just the introduction. And I know that you have a lot of experience with regulatory, and we'll be tapping you for some guidance on how we should continue and engage with the agency. But we're very, very interested in doing that.
We actually have had some interaction, and it's actually very meaningful. I need to remind myself of that. We did get it. We just haven't shared this publicly yet, but since we're amongst friends, we filed for rare pediatric disease designation for CMT1A. And that's actually a relatively big package, more than I was accustomed to. And that's where you're convincing the agency that this is a rare disease, it's in pediatric population, and that you've generated enough data that it makes sense for your therapeutic product to qualify. And we, about 10 days ago, maybe I can look at my email, we were granted our PD. So that's actually a very big deal, because just to share another anecdote between friends, not even all of the investors in the investor community are fully aware that this truly is a pediatric indication as well. And that does carry with it certain regulatory incentives. So I share this information with my colleagues as well. I think it's helpful to everybody.
Awesome, thank you.
I have a question.
Yep, where are you at?
Over here. So much, whoa, that's loud. There's so much attention in small molecule
right now in CMT. What in the world is a small molecule, and why does it matter in CMT?
It's a really great question. Go for it.
Small means literally a low molecular weight. So most of the things you're familiar with as drugs would fall into the category of small molecule, as opposed to an oligonucleotide or an antibody, which is a much larger molecule. And so much of the pharmaceutical biotech industry in the past has been based on those small molecules. So there's a lot of familiarity with how to go about that development. The newer modalities is a newer approach. So that's what we mean when we say small molecule.
Yeah, follow-up question.
Oh, yeah,
go for it, Rob.
I have two questions that are targeted to the whole panel. So the first one is, what can be done at the different stages of the development of a therapy, or what are the major roadblocks you've faced? What can be improved along the way? And then the second one is, it's very encouraging that there's companies that are pursuing the same strategy actually and kind of validating each other's work. But CMT is a very heterogeneous disease. It's a spectrum, and it's not a one shoe fits all. Does that affect the competitive space in CMT, that there may be several therapies coming onto the market, say for CMT1A? So one, competitive space. Second, what can be done to be improved, the translation of therapies?
Well, I think in terms of competition, I think, patients, I think you want that. I mean, you that's exactly what you want. You want competition. You want to make sure, one, that you have options, and two, that you get the best and the safest therapy you can get. So, I mean, as patient, that is exactly what you want. And I love that the CMTRF are, encouraging different mechanism of actions and, different strategies, even for the same targets. I mean, we're sitting here in the same table, and that is amazing. That is exactly what you want. Mm-hmm. So I think it's very positive.
And best might have a different meaning for different people. There'll be a spectrum within 1A. There'll be folks who respond better to one versus another. And so I think best will not be the same for everyone. So bringing multiple therapeutics forward, I think, is a good thing.
Drug development in the end comes down to the label. It's what's in the package insert that says what this medicine is appropriate to treat. And a big question is that going to say CMT, which is a clinical diagnosis, or is it going to specify a population based on a second test? So, for example, a genetic test that would define a subset of CMT. And I think that's well said. Competition is good. You want medicines of both kinds directed across the entire set of disorders.
Yeah. To kind of address the very first question that Rob said is a little bit about roadblocks. And I really appreciate that, Sylvain, you were very honest whenever you were talking about HDAC inhibitors and some of the roadblocks that you've had. Can you expand upon that a little bit and like how you're going to overcome them to get it to the clinic?
So yeah, the limiting factor for the HDAC6 field has been the toxicity. So it really not the target of antiviral has a toxicity problem. There's nothing toxic with the target. The biological target as HDAC6 is a compound, a chemical matter. And a lot of companies have been pushing advancing drugs to the clinic with failure due to the side effect they've seen, did in vitro and in vivo. And in patients, the third generation that has been still approved for oncology purposes has some hematotoxicity. So the new generation of drugs that has been, I would say, developed so far, we are targeting chronic disease, and the safety should be clear-cut, not a single side effect for such chronic disease. It was not the case for oncology patients that have, I would say, a lifetime difference. And so more of the company have been focusing on that, clearing the path to safety, whether or not have a good safety package, but also large efficacy at low dose. The goal is to have an oral drug, so an oral pill. And so if you have an oral pill, you need to be really safe because it's going to the body everywhere. There's a lot of, I would say, challenge in PNS and CNS disorders, or drug, for example, is a peripheral restricted, is not going to the brain, but there's a lot of compound targeting HDAC6 that also going to the brain, so are brain penetrant, and for targeting other kind of diseases, so ALS, FTD, Parkinson's disease. So the challenge will be different. The challenge of penetrating the brain will be one challenge for such drugs. But so far, that has been a lot of effort in designing brain penetrant drugs targeting HDAC6. And as of today, not a single company has been in the clinic with such drugs. So there's a hope. And that's why the pharmaceutical industry is so much interested by the target itself, because there's a broad of indication, but you need to have a good safety package for that. And that's why the most effort on this field has been cleaning the safety, because there's a lot of efficacy now proven.
Thank you. Rachel, do you want to add anything about roadblocks and overcoming them?
I think when you're dealing with a one-time, or not a one-time, not to be repeated for a long time therapy, there's questions that we ask ourselves and that the patients and that the regulators ask about durability. So you have to think about how you're going to tell that story, because nobody, well, you might say, okay, how do I know this drug is going to work for 10 years? I don't think the right answer is, let's watch an animal for 10 years, or let's watch 6 people for 10 years. That's not the answer. So we need to think about the data that we generate that's going to make us comfortable enough that we have comfort around the safety and some signals around the durability. So durability question, I think it's not so much a roadblock, but it's something that you need to think about strategically, with the assumption that you're not going to answer the question in black and white, because it's a bad idea. It's going to take too long. And then the other roadblock is these products are just very expensive to literally manufacture. And because the CMC, chemistry manufacturing controls, that you have to show to the agency that what you're making is the same thing every time. And we're talking about viruses here, and that's a very large molecule. It's not a molecule, it's gigantic. You end up having to literally consume a lot of your product to show to the regulators that your product can be characterized. So you need to have a strategy thought about ahead of time, how you can be most efficient with your product. And then in parallel, there is hopefully going to be some innovation that this can be done cheaper. The third roadblock, which is, I think, where the partnership comes in, somehow people, and these are everybody except for maybe us, don't always appreciate that CMT1A is a very serious disease, and that interventions can be justified in this disease because this is progressive, and that people do not want to lose their ability to walk or jump or use their hands the way they always could, or be in pain. So I do think it's very important that we continue to think very carefully about making sure that the regulators and just the broader community starts to appreciate that we're addressing a very serious disorder. So in a weird way, the resiliency of this population is a little bit of a roadblock, because when I walk to everybody and I'm like, how are you? Everybody tells me I'm fine. You need to start telling me you're not fine. So
more complaining.
Yeah, more complaining.
Yeah, we've got some time for more audience questions.
I have a question about the durability, and keep talking about 10 years and be able to predict 10 years. But so many drugs that have been put out, like statins, your COX-2 inhibitors and things like that, that after 2-3 years, people saw they had all kinds of side effects that build up with time, and they kind of manage it. Doctors will pull them off of it, maybe try something else. I would think, is the FDA setting the bar too high for what you're doing, or is this, as a community, you're looking at what to do 10 years from now, and it's not a requirement?
That is a great question. It's very contextual. So as an example, there was a gene therapy in hemophilia put out by BioMarin. Everyone was expecting it to get approved a few years ago. And it ended up surprising us. Roctavian, it only ended up getting recently approved. It got rejected because there wasn't enough durability, because there were other drugs in hemophilia that worked very, very well, as opposed to Luxturna, which was for a very rare form of blindness, where they didn't need to show that type of durability as the same bar for the hemophilia. So partially answering your question by saying it's contextual and it needs to be part of what you're addressing and the degree of unmet need and what the other options are.
So I would say because there are no drugs for CMT, exactly, that if you're first or second or third in the market, especially if you're at targeting different mechanisms, that's probably durability shouldn't be something you have to worry about initially. Or am I missing something?
You're not at all. We feel pretty comfortable that the animal durability that we're generating, that others are generating in terms of a class, is going to be adequate. I very much agree with you.
Okay.
Also, compared to the drugs you just mentioned that were treating huge populations for many years before those side effects were found, often that's a very negative outcome. There are drugs that are withdrawn at that point because the side effects turn out to be worse than the benefit. But in this population, very likely the FDA will actually lower the bar. The numbers of subjects you need to complete a data safety base to get the drug out to patients will be lower than it is for those large populations, and they will definitely tolerate a little bit more risk on that safety vigilance that will occur after authorization.
Which is why I was questioning why the focus on durability. Because most of you all talk about it when it doesn't seem like it's going to be a bar for approval.
Depends on the duration of your clinical study and the dosage regimen. If you're talking about a treatment that's a single administration versus a continual administration over the entire course of the study, I think that's the difference there.